Hello, everyone, and welcome to our first Mastermind Masterclass. My name is Candace Chapman, VP of Marketing here at Genomenon. Thank you for joining us for today's live session. Wherever you are, we hope you're safe and healthy and wearing pajama pants. This masterclass will focus on one of the latest features added to the Mastermind Genomic Search Engine, which is the ability to search by phenotype. This is a powerful feature that even during our rehearsal this week, we were exploring more applications for, and I can't wait for you to see what can be done with it. Searching by phenotype is a professional feature, so if you currently have the basic edition, some of this may not look familiar to you. But during the demonstration, you'll see many features that you may not be aware are available in Mastermind. So this is a great time to get reacquainted with the full functionality of Mastermind and see if you and your organization can benefit from Mastermind Professional Edition. It's our goal to make Mastermind the first resource you use when interpreting variants, and I hope that this deep dive masterclass will help you get the most out of it. Let me cover some housekeeping items before we get started. You can submit questions in the question box at any time during our time together, and we'll have a Q&A session with the founders after the demo. Today's masterclass is being recorded, and a link to the presentation and the Q&A transcripts will be emailed to you Friday or maybe Monday. So I'm really excited about this webinar because we've gathered not one, but two of our amazing founders to discuss Mastermind in depth. We also have an MVP from the data science team to walk you through the demo. Introductions are usually pretty boring, so I thought we'd change it up a bit, and I asked our presenters to share something interesting that you might not know about them. Our first presenter is Dr. Mark Kiel, co-founder and chief science officer. A paper Mark authored disproved the immortal strand hypothesis, so that's pretty cool. You can Google it. Mark, you'll be sharing a brief Mastermind scientific update. Next up is Steve Schwartz, co-founder and chief technology officer. Steve has a competition, hey Steve. Steve has a competition racing license, and he races when he has the time, which is almost never since the birth of his two daughters, but I think I know his priorities. Steve will be sharing a brief Mastermind technology update. And then we have our SHIELD, or our special handler for information and essential logistics development, Diane Nefsey. Diane tends her own tree farm with her husband and her toothless cat. But here's a few more interesting facts about these guys. What are the odds that both Mark and Diane have twins, identical twins? And Mark named his children after characters from Greek mythology. Diane named her pets after characters from Greek mythology. But not to be undone, Steve claims that he has books about Greek mythology. So, but enough about the introductions. Let's welcome Mark for the scientific update. Take it away, Mark.
Hey, thanks, Candace. And thanks everybody in attendance for joining us for what is the first in a series of Mastermind Masterclasses. I'm going to give a little bit of a nostalgic view back in time of where Genomenon started. I'll just spend 2 or 3 minutes, and then I'll pass it over to Steve and Diane to go over the practical aspects of what we wanted to showcase here on the webinar. So many of you may not know, but Genomenon, which actually is Greek, it means born out of need, was started about 7 years ago when I was an MD PhD, frustrated that I was constantly needing to go out to Google to get the genomic information I needed to either diagnose my patient caseload or otherwise to complete the discovery work that I was doing in the research lab. And since that time, we've accumulated a very robust and increasingly large user base that numbers around 7,000 across 100 and more countries and continues to grow at a near exponential pace. And at Mastermind, the software that we're going to be discussing today is increasingly becoming the go-to resource for genomic evidence for both clinical workflows and for making pharma discoveries with some of the work that we're doing with pharmaceutical companies and biotech companies. And this ability to grow the user base and to become the dominant force in genomic sequencing interpretation is due in part to the advances that we're making, such as the one that we're going to showcase today, the addition of the human phenotype ontology, or phenotypic terms, which are particularly valuable in solving otherwise mysterious rare disease cases for patients for whom there is sequencing information. So as Candace alluded to, I'm very happy to be sharing the spotlight with my co-founder and friend, our CTO, Steve Schwartz, and one of our earliest employees, and somebody who I don't think gets enough credit for in many ways keeping Genomenon running smoothly on a day-to-day basis, Diane Nefsey. Very happy to be on the webinar with both of them. Steve is going to walk through the technology at a high level that is driving this new development, this new feature that we're adding into Mastermind. And Diane is going to walk through several use cases, a couple in the user interface and a couple also in the API. And then Steve is going to round out the conversation by showcasing some of the powerful capabilities and value that can be elicited through maximal use of the API through some scripting. So without further ado, I'll pass it over to Steve, and I'll be rejoining the group here during the QA session toward the end of this webinar. So Steve?
Thanks, Mark. Yes, so let me pull up some slides here real quick. Here we go. All right, can everyone, well, I guess I can't hear anyone. I'm used to conference calls here. Okay, so as Mark said, I'm going to focus on the high level of the technology, try to make it not too boring for everyone. The technology behind Mastermind really empowers us to provide two services to our users. The first is organizing and indexing the entirety of the medical evidence, making it easily searchable. The idea being that it gets the most information to you the quickest so that it can get out of your way and you can continue, you know, if you're on the clinical side, making a diagnosis, if you're on the pharma side, doing research. The second is identifying every genomic association that's supported by that medical evidence. So the genomic associations that I'm talking about stem from searching the entirety and indexing the entirety of the medical evidence for genomic entities, what we call genomic entities. So those are genes, variants, diseases, phenotypes, which we're talking about today, as well as categories such as AC and G and AMP criteria, and therapies, which will come at a later date. So the idea here being that when we find every one of those entities throughout the medical evidence, we can then identify the associations between any entity and any other entity in that graph. And from those associations, that's how you can start to identify hypotheses and move those hypotheses into insights. So a standard use case might be a patient has a disease and you need to find a therapy. That's great, but any hypothesis or any association directly between disease and therapy really is just a hypothesis until you can start to identify and understand the causative link between that disease and that therapy. So in the context of genomics, that evidence tends to be genes, the function of genes, and the variants within those genes. And so you can see here, every time that we can add a causative or an entity into that causative link, we add a piece to the puzzle and we understand that connection much better. So the strongest insight that you can find is where you can connect, for example, a disease to a therapy through a specific gene and variant categorized by phenotypes and categories. And so that's what we're really enabling our users to do here by adding phenotypes to our ontological structure and starting to draw those connections. And so an example of how you might do that in a clinical context would be you have a patient that has, say, 3,000 to 5,000 variants that you've managed to filter down from a whole exome screening, and you have a set of phenotypes on the right, And what you can start to do with our search engine, or even more powerfully with our advanced API, is you can start to iterate through every possible association, not just between one variant and one phenotype, but between any and all of the variants that the patient has and any and all of the phenotypes that they have. And you can start drawing those connections much more easily, again, using the medical evidence as the basis for informing those associations, so that you have strong evidence that you can then put into a report and make conclusions based on. In this example, we're using variants, connecting them to the phenotypes from the medical evidence. And then in those same papers, we can further then look through the papers and see what diseases or what therapies did those papers also talk about, to help automate the process of diagnosing a patient or generating a case report, or again, on the pharma side, starting to automate the process of drug discovery and drug development. So with that, I will now turn it over to Diane, who can show you how this phenotype data actually looks in the API and the user interface.
Excellent. Thank you, Steve. And while I am working on sharing my screen here, I'd just like to remind you that not all of the features that you'll see today are available to basic accounts. And if you would like to test the features that you don't have access to, just send us an email. We'd be happy to help. So today I'm going to walk you through some user interface Mastermind examples that will allow you to discover a set of genes or diseases associated with your patient's phenotype, or even a combination of phenotypes. So, in this first example, say your patient has a phenotype that you suspect has an underlying genetic cause, and you'd like to find a candidate gene for testing, and not just a candidate gene, but literature evidence for it. In this first example, we have hyperpigmentation of the skin. And if you see this drop-down list, not only can you choose the phenotype, you'll have the typo-free phenotype. And the other one that your patient has in this example is hamartomatous polyps. And right here we see the phenotype and the proper canonical HPO term. And we have both of these together in our patient. We'll click search. And right away you can see in our results that the STK11 gene is the first hit on the list with 81 articles. And that is expected because these are symptoms for Peutz-Jeghers syndrome. And in our next search, we will show you that you can search with either one or two phenotypes, or both of them combined. So your patient, for example, has a family with a variety of phenotypes. The first one being Ectopia lentis. And we'll have the phenotype Ectopia lentis. And the second phenotype we have here is arachnodactyly, also as a phenotype. But not all of the people in the family have them. So we will click the OR to toggle between and OR. And as expected, the genes on the list are organized by the fibrillins and elastins first. And we can use this interface to look more deeply at the abstracts, titles and abstracts of the papers here. So right now the default sort is by relevance, and if you click on any paper, you'll see that by default we have genes and variants highlighted, but you can switch to phenotypes, and you can see that in the full text, anytime that there's a phenotype mentioned, you'll see that highlighted and a sentence proof is shown to you. So one more example we have for you today is something that would help you maximize specificity for your patient. So say you have a patient with hyperphagia. You'll see that not only does it correct spelling for you, but it also gives you the proper canonical HPO term. So we have polyphagia here. However, and as you can guess, we have a lot of articles that may or may not help you for your specific patient. But in this case, you know that your client has some sort of chromosomal aberration at the 15q locus. So if you enter that in as a text term and search for it, what this will do is this will bring up all the articles that mention 15q in the full text. And this brings up all the genes associated with the Prader-Willi locus. And if you click on them, we can see that it brings up papers for Angelman syndrome and for Prader-Willi. Very cool. So with those out of the way, I'm going to show you some API examples. So that's how a human would do it, and this is sort of a demonstration of how you would automate this. So just like this last example, if we fetch our API token and our HPO term of interest is hyperphagia, and we submit that query, we can see that all of these outputs are all labeled, and we have the proper HPO term. And so we can take that and we can go to our disease endpoint. We can plug the HPO term in right here, and you can see that the API token carries over. And this will give you the output of all diseases associated with that HPO term. And this is just page 1. And for example, we can flip through to page 2. And here on page 3, we have Prader-Willi syndrome. So if you want to find articles associated with that, we'll highlight that. We'll just go to the articles endpoint, plug that in. And in our suggestions endpoint, we can grab our HPO title, go back to articles, put that in. And here we go. We have a full count of how many articles are associated with it. You can scroll down and see all this information here. But if we're only interested in one specific paper, we can take that, go to our article info endpoint, plug it in, and not only will we see the title and the abstract, but as we scroll down, everything that was discovered in the full text will be shown. HPO terms, and all the way down here at the bottom, we've got all of the genes that were found in this paper, as well as the gene variants. Let's see, finding you an example of the variants that we found in here. And as you can see, this is quite a meaty paper.
Diane, it's likely that there aren't any in that paper, but that's exactly right. The article's info endpoint showcases all of the indexing results for that paper: phenotypes, genes, gene variants, title, abstract, and all the associated metadata.
Right. So you can imagine doing this as a single query, and all of these steps would be a fraction of a second to complete. And if you do this in bulk, well, that would be even more powerful. So we'll show you how to discover literature evidence here that provides a link for your client's phenotypes and genetic variations. And as before, we have, just as an example that I already have available for you, if you say that we suspect the Notch gene and your client has one specific disease, you can search and you will find this very handy variant diagram plot that will help you discover just where in the gene you would search for any suspected variants. And if you have the same gene and you choose a different syndrome, you can see that the variant plot is very different. So we're going to go through one more API example for that, using Hajdu-Cheney syndrome as an example. So if we have the HPO term, you want to find out the HPO term for acroosteolysis, which is a symptom of Hajdu-Cheney. We'll find out that this one in particular, phalanges of the hand, is one symptom of Taj-Duchenne, so you will copy that. And we'll go to our variance endpoint. We'll put in our HPO ID and the suspected gene. Submit that query. And we have a handy list of all relevant variations, including the article count. And if you're interested in those articles in particular, we will go to the articles endpoint. We'll put in our variant interest. We will grab that HPO term. Go. And we can see a comprehensive list of all those 8 articles associated with the variant of interest and your client's disease. And as before, if there are extra pages, we can flip through those. So as before, the Mastermind interface is great for a visual overview of these features, and the real powerful features of the API allows you to do all of those things in bulk. And with that, I'll hand it over to Steve, and he might give you a more in-depth explanation of our API.
Awesome. Thank you, Diane. So yeah, so what you just saw was a demonstration of the UI and the individual API endpoints. One thing that really starts to make the API powerful, as Diane alluded to, the user interface is extremely useful when you're doing sort of ad hoc investigation or discovery work, trying to figure out what exactly it is you're looking for. Once you figure that out, the API makes it much easier, much faster to start automating that process across use cases. You can integrate it into your existing pipeline, and you can really start to do much more in-depth analysis that isn't very practical or realistic for a human to do in real time. So one example of that, as I showed earlier with a graphical representation, is analyzing a lot of variants and a lot of phenotypes at one time. But again, going back to the association visualization that I showed, there's a lot of different entities that you can analyze on in order to start to automate the production of that output. So what I was going to show you here real quick is an example script that we put together that looks at a collection of variants from a given patient with a list of phenotypes for that patient and does an aggregate analysis across all of the medical literature to start to find any multiples of variants or phenotypes within that patient that tend to be co-cited within the medical literature, meaning papers that talk about more than one of those variants with more than one of those phenotypes at the same time. And so, again, this is a technical demonstration of an API, which doesn't tend to be extremely exciting, but I will try to make it as interesting as I can here. So what we have here is a script that I've written that takes a list of variants as input and a list of phenotypes as input. In fact, let me show what those lists look like real quick. So I just have a few sample variants that I put together here that look like this. So you can see it's just a collection of several variants across several different genes. These aren't necessarily related to each other. It's just a bunch of random variants that I threw into a file. And you can see that the script is easily able to accept the variants in whatever format that you have, whether it's the protein nomenclature or cDNA. It can also handle RSID, genomic coordinates, and IVS nomenclature. And then I also have a set of phenotypes for this patient, again, made up as well. And so what this script does is it takes that list of variants that I just showed you, as well as that list of phenotypes, and it starts analyzing them in aggregate. So the first thing it does is it finds the canonical HPO terms that match each of those phenotypes in the list. Then it will search through each variant, finding every article.
Maybe while he's rejoining, I think he'll be able to come back, and hopefully he'll get a screen that illuminates the results of that script. But I'll speak from a clinical perspective of the benefit of that script. as Diane alluded to while she was showcasing the web interface, and how you can iterate through those queries to amass all of the insight and information that you're seeking around your clinical case. What Steve is highlighting is the ability to do that all at once for any given patient's sequencing data, with all of those results. Once you've configured the API calls and the way that the data gets output, Steve is going to hopefully show his screen and showcase the fruits of that automated workflow to give you immediate insight, no matter what the phenotypes are that are input, no matter what the clinical circumstance, no matter what the list of variants. The script that Steve was running that was built on the APIs, I like to say, automatically assembles all that data and prioritizes the insights that you need to very quickly and very accurately, sensitively make the appropriate diagnosis for your patient based on the information content throughout the entirety of Mastermind. So Steve, thanks for jumping back on there. This is the output screen that I was suggesting he would show. And again, this is particular to the example that Steve ran and the way he configured the script, but it should be apparent to you how valuable this collected information is for prioritizing the results and, again, getting immediate and keen insight into the nature of your patient's disease based on the clinical circumstance. So Steve, if you're on, again, I'll turn it back over to you.
Yes, thanks, Mark. Thanks for rescuing me there. Yes, so this is an example of the output from the script that I was just showing. In this case, it found 3 different phenotypes that have been co-cited in 3 different articles, matching 2 different input variants. So you can see the 3 phenotypes that it found in the articles. Here are the 3 PMIDs, and these are the 2 variants that those 3 articles, all 3 mentioned. And then to help start to help you automate the process of finding a diagnosis, it will also list out the diseases that were cited in those 3 articles. And then if you scroll down, here's another output where it found 2 different phenotypes within this large set of PMIDs, also co-citing these 2 variants. And here's the larger set of diseases that those found. And then you can keep going down and see the rest of the associations. Again, this is just the example input that I had. We've run this for many more variants, between 3,000 and 5,000 variants and 15 to 20 phenotypes per patient, and it can still automate the production of this kind of output. It's an API call, so in this case, I happen to have it outputting in an indented file that makes it a little easier for me to demonstrate here the kind of information that it's outputting, but it also can output in any format that you would like, such as CSV, a VCF file, or even just directly putting that data into your own pipelines database for you to interpolate the results into your own pipeline, your own user interface for your variant curators or for your research team. And so again, this kind of shows the power of the API and the kinds of things that you can start to do once you automate the multifaceted association identification between genomic entities for a given patient, or again, for drug discovery or any number of other use cases. So I will pass it back to Candace now.
Thanks. Steve, can you hear me all right?
We can.
All right. So it's time for questions and answers. Thanks so much to Mark and Diane and Steve. Are you seeing my screen?
Yep.
For the Q&A? All right. This would also be a great time to share your feedback with us by filling out our new 2-minute mastermind survey at genomenon.com/feedback. So let's get to some questions. Thank you everyone for sticking with us through our technical difficulties, and we will have the recording if anyone had audio difficulties. You know, hopefully the recording will be able to hear what you weren't able to hear. Kent asks, does your search engine enable finding connections within and across other species like mouse and mouse to human?
Steve, I don't want to be rude to each other. I don't know if you want to take the first one and I'll take the second and go in that order.
Sure. Yeah, it doesn't matter to me. It does not.
Uh-oh. Maybe Steve.
Currently do this for other species, but we have built the platform with the ability to do that. So we have done it on a case-by-case basis with some customers in the past.
Yep, I'll say that the gene synonyms for human genes often always include those for lower species, like even yeast. And certainly there's a lot of parity across the mouse and rat and other standard research tool gene names. And the hexagon that Steve showed, bringing all those associations together, would certainly be true across species, absent the variant, which is much more species specific. And there are ways to impregnate in your API query some of those species names to specifically pull out that information as you please in your API calls. So that was a great question.
Okay. Birgit asks, how many HPO terms can be entered at a time?
As many as won't annoy the user. So in the user interface, you can collect as many as you'd like. I like to say where we showcased how automatable the API is, the user interface can be programmed to include those HPO terms because it has URLs that are, it's called deep linking. Is that right, Steve? It's got the actual term in the URL. So if you have a workflow and you're either savvy with Excel or you have an informatic group, you can pattern all of those HPO queries into a link that you then click into Mastermind for any given gene. And Steve, I don't know if there's a limit. As I said, I think it just, you could probably put in the 100 and it would still execute.
Yeah, there's an answer for the user interface and an answer for the API actually. So as you said, Mark, there's not an explicit limit. I actually don't know. I haven't personally tried stressing the system to see, you know, if it's 100 or 1,000 or 10,000 HPO terms. I'm actually not sure at what point the application starts to bog down and not be able to complete the request. Theoretically, the more HPO terms you put in, the slower it should get. But in our day-to-day use and in all of the use cases we've seen with the customers that we've been using this with so far, we haven't seen any practical limit on how many they can do. On the API side, there's actually the script that I just showed you and the output from that. We actually built that script with some sort of HIPAA considerations in mind. We wanted to limit the amount of information that was actually being sent to our server in the first place. And so the script that I just showed you doesn't query our server for HPO terms at all. What it actually does is it finds every article for every variant, and then it looks through the article info endpoint that Diane showed that shows the results of everything we've indexed in the article, and it pulls out all of the HPO terms from that output, and then it mashes them all together and then does an intersection operation between that and the phenotypes that you passed into the script. So the phenotypes that you passed into that particular script, again, it was written in a way that it doesn't send those to the server. So at least for that script, there's literally no limit to the number of HPO terms you could pass into that script because it's never even sending them to the server anyway. It's just grabbing all HPO terms from all papers that it queried for the variants and then doing the filtering on HPO as a post-processing operation on your own machine. So again, on the UI, I'm actually not sure what the limit is. We haven't run into it yet. And on the API side, it can be written in a way to where the phenotypes don't even hit the server.
I will say one caveat to that is it's probably not a technical concern like Steve was describing, and more of a sensitivity specificity concern. So the more entries you put, if you have and, the less and less likely you're going to find all of those in any given article. So that's something that you should be very mindful of. And if you toggle to an or and you add a dozen, you're going to start to get non-specific results. And so that's why I said it's to the user's tolerance, and it bears just sort of testing empirically what kind of results you get with what types of phenotypes. Great question.
That's why this is the master class. I mean, you guys are geniuses. It's amazing what you have done with Mastermind. Next question from Simone. What determines the order of genes on the list in the result of the search?
I'll take that, Steve, real quick. It's prioritized by number of articles in its sort of base presentation. So with Diane's example for Peutz-Jeghers syndrome and STK11 being present, you saw P10 being present below that, I think, with 80 some articles, given that it does have pigmentation challenges and hamartomas. STK11 had 50% more results, and so it was prioritized up front. And then there was a very steep drop off after that. So in its simplest version in the user interface, the results for gene matches when phenotypes are input is just on the sheer burden of results. We obviously do have very sophisticated capability to prioritize the results on the variant side in that list of references that include variants. When you add any key terms, those key terms and how close together they are to the variant or gene that you're searching for, as well as how frequently those key terms appear throughout the page, that is a very high-level view of how sophisticated we have prioritization algorithms present those article results to maximize the likelihood that the first 5 or 10 papers will be papers of merit that you render a diagnosis with. And especially in light of what Steve was able to show, sky's the limit on all the ways that you want as an informatician to prioritize the results based on the collected API returns that emerge from your query.
Yep, and I'll add, since this is a master class, I can add a little bit more depth to that as well. When talking about the prioritization of results within Mastermind, the UI and the API, I often will sort of dissect the problem into two main components that make it a little easier to understand. The first is, well, to understand both components, first understanding that the associations and the results that we return are from the medical evidence. So they all stem from what has been studied and cited in the literature. And so understanding that, the two components that we often look at when you're thinking about, firstly, the order of article results for any given query is determined by a sort of static association strength and a dynamic association strength component. So the static association strength comes from characteristics of each article in and of itself. Things like what journal was it published in, what was the publication date, what is the impact score of the journal, the citation count of the article, things like that. So those are sort of characteristics that follow each article around, no matter what the search is that you're doing that results in that article being present in the results. And then there's the dynamic component, which is how relevant is the article to the thing you just searched for. And so that's where characteristics such as Mark was describing come into play, meaning, you know, if you search for a variant in a phenotype, how closely were those cited in the article? How many times was each of those cited in the article? How many times were they cited in the article relative to other entities in the article? So for example, was my variant mentioned once in the article, and the article also mentioned 300 other variants because it was a GWAS study, or was my variant mentioned 5 times in the article and only one other variant was ever mentioned in the same article, meaning the article was about my variant? And so, you know, things like that, you know, how strong is the evidence for each term that you search for? And then how closely related are those 2 or more terms in the results. And so that's how we prioritize articles. And then you can see how our capabilities become more and more sophisticated to, for example, rank the genes, as you asked about, the genes as results to your search query, because a lot of the strength of evidence for the genes stems from the articles. And so we can rank them, for example, on how much were the articles that inform that gene association relevant to your search. Another fact were cited in those articles that inform that gene association. And it keeps going. There's so many factors that we use in that prioritization. It's something that we're continuing to refine and improve.
Great. So Yaron asks, can you put parentheses between the search terms, i.e. use a combination of and or?
That's a great question. Not currently. Right now, we've tried to build the what we refer to as the Boolean search capability. So the capability of searching for more than one term within any given entity. So a regular search would be up to one term of each entity type. So for example, a gene and variant and disease and phenotype would be a pretty complex basic non-Boolean search, because it's just this and this and this and this. The Boolean search comes into play when you might have more than one term for any one of those. So for example, this gene and this gene and this variant or this variant or this variant and this phenotype. So our current Boolean search has the parentheses implied, and the way that those parentheses are implied is they're around each entity type, and then you can change the and or the or within any one entity. So for example, you can search for, in parentheses, gene A and gene B, and new parentheses, variant A or variant B or variant C or variant D, and parentheses around diseases, parentheses around phenotypes. The ability to then change those parentheses and, for example, have gene A and variant A, or gene B and variant B, or, and sort of move those parentheses so that they're around combinations of entities is not something that we currently have. We have discussed it, but we haven't yet had any users or customers reach out to us that it was an absolutely necessary component of their pipeline, or something that they had really even started investigating doing yet. All of that said, you can do that in the API, because in the API, you can just put ors between everything, and then you can do whatever combination of logic you want to do, and wherever you want to place the parentheses you can around the analysis of the results as a post-processing operation on your own machine in your own code. So it is possible to do with the API as a post-processing operation.
I was going to add that. Seems like great minds think alike.
All right, Carol asks, if the phenotypes entered by the user don't include the output phenotypes, what will you see?
If the phenotypes entered by the user don't include the output phenotype.
I mean, I'm assuming that
question is if this question is in relation to the script that I showed.
Probably right.
Yeah. So in the context of the script that I showed that uses the API to do the variant phenotype association and identify diseases, I think the scenario we're talking about here is the variants and the phenotypes that you input don't result in any articles that mention those variants together with any of the input phenotypes. What that script, that's a great question actually, what that script does, the output that I showed you is only the summary file that the script generates. It actually generates 5 different files. It generates 2 summaries. One is a really high level summary, and then one is a very detailed summary. And then it generates 3 or 4 CSV files that list all of the results by article, all of the results by variant, all of the results by gene, all of the results by phenotype, and all of the results by disease. So however many files that was. So what will end up happening if none of the variants are cited together with any of the input phenotypes is the summary file will actually show no results in the section that has the variance with the phenotypes. Above that, there's a different section that will look for any of the variants that were co-cited together in articles absent phenotypes, absent in any phenotype matches. So you'll still see the results of any variants that were cited together with each other, what those PMIDs were, and then you'll see a list of all the phenotypes and diseases that those articles had, regardless of whatever you're in. So you will be able to see, okay, none of my phenotypes were.
Steve, we're losing you there, but I can summarize what I'm imagining he's based on. It's tiered, right? What you, I forgot, or I didn't pick up that you were talking about the script, but if your specific input phenotypes are found, that appears front and center, maximal efficiency in finding the results that you had asked for. If none of those input phenotypes are found, what Steve was getting at was the APIs and the script assembles all of the phenotypes irrespective of what your input was and puts that at the second tier, which if the first tier is null, you'll see immediately emerge. So I like to say there's those two phases. One is recovery of an exact match or even a partial match of what you input. And the second phase is discovery of things that you didn't even know you wanted to see that comes out of that vast association network that Steve described and showcased the script invoking for any given variant data set for any patient.
Okay. We just have questions keep coming in, so I'm going to keep asking till the top of the hour. Like I said, there will be a recording of this, so if you want to listen to it at your leisure or look at the transcript, we'll flesh out the questions as well so that anything that was kind of missed here will be caught in the transcript. But Paula asks, how does this tool manage the issue of having many different transcripts for a single gene? Papers do not always have proper information on which transcript is being used for naming variants.
They almost never do. That's right, Paula. Steve, I'll turn it over to you, but I'll answer succinctly that Mastermind handles those handily. We have something called genomic language processing that is responsible for both the indexing of the content as well as understanding what the user's input is. There's a lot of complexity there that belies my short answer, but Mastermind is genomics aware of all these transcripts, attendant legacy nomenclature issues, all of the nuances of the genetic code and the complexities of the human genome. And that's one of the value propositions of Mastermind, is that you needn't have to worry about or think about those things because our infrastructure has already done that consideration for you and presents the results appropriately. So Steve, I don't know if you want to speak to what the user experience is of that they get the benefits of that maximal sensitivity. It's cutting in and out. So let me suffice it to say, I think Paula had asked that question, that the indexing recognizes all of the transcripts for any given gene. And when the authors haven't declared what transcript they're speaking in terms of, Mastermind recognizes which of those transcripts the variant may be associated with. And if the authors didn't declare a specific transcript, we maximize the sensitivity of those results by associating it with all of those transcripts that are appropriate based on the cDNA mention or the reference allele at that protein position. And then, as Diane showcased in one of her examples, we show the sentence context where the authors describe those matches so that you can then be the judge. Is this my specific result? Or do I disagree that this is the very variant that I was looking for, in which case you can just very quickly move on. And I think what Steve was interested in getting at was when you perform a search at the cDNA level for a specific variant, one of the mechanisms that we have in place to prioritize those results will be aware that you're looking for that specific cDNA match. And in addition to prioritizing according to that match, will showcase very quickly in that lower right compartment where the sentence context is mentioned what exactly the author said. So you can rapidly toggle from result to result and just be sure that you're getting the most specific result depending on your specific clinical concern, whereas other circumstances warrant that you benefit from seeing all of those results irrespective of the specific cDNA match. And so Mastermind allows you to play both sides of that fence.
All right, I'm going to ask one more question. And since this is a master class, I'm going to hit you with a hard one, all right? Are you ready?
You get your microphone ready.
Matt asks, how well does Mastermind prioritize genes and variants in complex, undiagnosed disease cases, or potentially a new genetic condition? Imagine a complex congenital condition with dozens of phenotypes across multiple organ systems and dozens of candidate genes or variants.
Awesome question, Matt. It gets back to the answer that I gave about data recovery and data discovery. So the recovery is when you've got phenotypes and variants that you've searched on. If there's any evidence in the literature, we'll pull that out and show you exactly what those references are. If what we're talking about is no explicit associations that Steve alluded to that very broadly unite the disease and the variant and the gene, etc., and the phenotypes, if that kind of information doesn't exist in the literature, you would default then to that discovery phase. And so where you're talking about complex undiagnosed cases, the power of the API that Steve showcased is its ability to draw on all of that information and aggregate it all for you in a way that you couldn't hope to do manually, and then present you the fruits of that work that was automatic in a ranked prioritized order. So whenever you're looking for things that aren't crystal clear, you should be able to tolerate some false leads. But when you have no other option, the API that Steve showcased and similarly configured APIs will give you the best opportunity to very quickly find among those false leads any real lead that will then get you to the eureka moment that I think you're seeking when it's an undiagnosed case. Diane highlighted a facile example with Prader-Willi, where you have 15q chromosomal effects. You can imagine if you've got a case where it defies diagnosis and you've got some abnormal chromosomal microarray results and some confusing constellation of phenotypes that don't form a syndrome in your eye as you're looking at it clinically. If you would input those results into the Mastermind user interface or the API, as I said, you may very well have that eureka moment based on the prioritized results that would otherwise take you probably many hours to days to comb through iteratively doing Google searches or what have you, if you're able even to find that information at all. So that is really the power of the script that Steve showcased in the specific version that he showed and in any permutation thereof, depending on your inputs and the types of queries that you're looking for. So your question was quantitative. I can't answer it quantitatively. Suffice it to say, though, that that's one of the powers of the capability of the Mastermind API is data discovery in that vein. So, Steve, I don't know if you're live on audio, but hopefully you agreed with my handling.
I think so.
Okay.
Yeah. You know, I'll also add that we've actually had quite a bit of experience, I think, with that specific scenario exactly, where we worked with one large company, for example, that was doing a trial of our API, and they ran a script very similar to the one that I showed, where the first thing they threw at it was a difficult, unsolvable case that they had for a rare disease. They sent the patient's information to several different labs and other providers, and I can see it. I can actually see the audio quality right here. Okay, it's green again. I'll talk fast when it's green. So they had sent it to several other labs that were unable to explain the combination of phenotypes that the patient was having. And then they tried it with our software, and it actually came up with a combination of 3 different variants that the patient had that when combined, there were 3 different variants, I think, across 2 different genes that when combined fully explained the full scope of phenotypes that the patient had and led them to a candidate diagnosis that they had never been able to find before. And that was just the result of the one-off trial that they did with us, where they tried to throw one of their toughest cases at our API. We've done similar trials with several other companies that have seen, in some cases, anywhere from 10 to 50% diagnosis rate on some of their hardest cases. So we've definitely seen, I think, the data shine in those kinds of scenarios.
Awesome.
Yeah, good.
Thank you, guys. Yeah, we're gonna run the rest of the questions by Mark and Steve and have them answer them in a physical format so that we can put them on our website. But I just want to thank you so much for joining us today. I hope you learned things that you can use in your daily work. And I'm very excited about our next Mastermind Masterclass. It's already in the works, and we'll be focused on a feature we'll be releasing very soon, which is searching by therapy. We think this feature will be a game changer for many of our users, and we can't wait to go live. So stay tuned to your email and on Twitter and LinkedIn at Genomenon for more info. And if you don't have a Mastermind account yet, go to bit.ly/Mastermind Masterclass to sign up, and there's no cost for the basic edition. If you'd like to talk to a member of our team about upgrading to professional edition, or you have questions about purchasing multiple seats for your lab, email us at hello@Genomenon, and visit Genomenon/feedback to share your feedback. And thank you again for joining us. We hope you join us for our next Mastermind Masterclass. Thank you to Steve and Mark and Diane, and enjoy the rest of your day.